Hello everyone, welcome to another exploration into the extraordinary capabilities of the human brain. Today we're delving into a fascinating topic, how we remember. From the intricate workings of the hippocampus to groundbreaking experiments in memory research, we'll uncover the science behind one of the most vital aspects of human cognition. Let's begin with a story that revolutionized our understanding of memory. Meet Henry Molaison, better known in scientific literature as HM. At the age of 9, Henry suffered a head injury that led to debilitating seizures. At 2, he underwent an experimental surgery to relieve his symptoms. The procedure succeeded in stopping his seizures, but it came at a significant cost. His hippocampus, a crucial brain structure for memory, was partially removed. Post-surgery, Henry was unable to form new memories. He could remember events from before the operation, but was trapped in the present, unable to retain new information for more than a few minutes. Imagine greeting the same person several times a day, as if meeting them for the first time. Henry's case became a cornerstone for neuroscience, highlighting the hippocampus's pivotal role in converting short-term memories into long-term ones. So, how exactly do we remember? Let's break it down. Sensory information from our environment first passes through the brainstem to the thalamus, which acts as a relay station. From there, it reaches the prefrontal cortex, where it forms our conscious short-term memory. But here's the kicker, short-term memories are fleeting. To make them stick, they must pass through the hippocampus, where they're processed and encoded into long-term storage. Here's where it gets fascinating. Memories aren't stored in one specific location like files on a hard drive. Instead, the hippocampus distributes different aspects of a memory, emotions, visuals, sounds, across various parts of the brain. For instance, emotions might go to the amygdala, while visual details reside in the occipital lobe. This decentralized system ensures efficiency and resilience. When we recall a memory, our brain must reassemble these scattered fragments into a coherent whole. Scientists call this the binding problem. Imagine recalling a walk in the park. The sound of birds chirping, the feeling of sunshine, and the scent of flowers must all come together seamlessly. The binding problem seeks to answer one of neuroscience's most puzzling questions. How does the brain reconstruct a coherent memory from fragments stored in different regions? When we remember an experience, such as a walk in the park, our brain retrieves scattered details like sounds, sights, smells, and emotions and integrates them seamlessly into a single, vivid memory. Scientists aim to understand how this process works and why it sometimes fails, such as in cases of amnesia or neurological damage. One of the most revealing insights into the binding problem comes from observing patients who have suffered strokes. These individuals often experience selective memory deficits, such as the inability to recall specific types of information, like faces or names, while retaining other details. Researchers like Dr. Antonio Damasio use these cases to pinpoint the brain regions responsible for specific aspects of memory storage and retrieval. For example, a patient might remember walking through a garden, but fail to recall the face of a companion. This suggests that the memory of faces is stored separately from other elements of the experience, and that damage to specific brain regions, like the fusiform face area, can disrupt the retrieval of those details. To investigate the binding problem further, scientists used advanced imaging technologies like functional magnetic resonance imaging, or fMRI. These tools allowed researchers to observe which brain regions activated during memory recall. The studies revealed that different parts of the brain light up depending on the type of information being retrieved. Visual elements activate the occipital lobe, while emotional aspects engage the amygdala, and spatial details are processed in the parietal lobe. Researchers conducted experiments where participants were asked to memorize complex scenarios involving multiple sensory details. Later, participants were shown partial cues, like an image of the scene or a sound associated with it, and asked to recall the entire experience. Brain scans showed heightened activity in the hippocampus as it acted as a hub, coordinating signals from different regions to reassemble the complete memory. The binding problem is critical to understanding not only how memory works, but also how it breaks down in cases of brain injury or disease. When the brain's ability to integrate fragments of memory is disrupted, individuals may experience fragmented memories, recalling isolated details without context, as seen in some forms of amnesia. False memories can also occur where unrelated details are misbound to create a distorted or entirely false recollection. Finally, memory loss may happen where there's a failure to access specific categories of information while retaining others. Understanding the binding problem could lead to breakthroughs in treating these conditions. For instance, targeted therapies could help stroke survivors regain the ability to bind sensory information, restoring the coherence of their memories. Research into the binding problem could have profound implications. First, artificial memory reconstruction. Imagine developing technologies that mimic the hippocampus's binding function, allowing us to reconstruct memories for patients with neurological damage. Second, virtual reality for memory therapy. Virtual environments could stimulate sensory regions of the brain, aiding in the recovery of specific memories by providing missing contextual details. And third, memory enhancement tools. By understanding how the brain binds details, scientists could develop methods to strengthen these connections, improving overall memory retention and recall. Now, here's a twist. Memory isn't just about the past, it's also about the future. Studies show that the same brain regions involved in recalling memories, like the dorsolateral prefrontal cortex and hippocampus, also light up when we imagine future scenarios. Dr. Kathleen McDermott describes this as mental time travel. It's the ability to project ourselves forward, using past experiences to predict and prepare for what's to come. This capacity for foresight is a hallmark of human intelligence, setting us apart from most other species. Now, have you ever thought why emotionally charged memories are so vivid? Emotions can amplify memory formation by altering the activity of genes like CREB, which is involved in strengthening neural connections. This mechanism ensures that significant experiences, whether joyful or traumatic, are encoded deeply into our long-term memory. Memory is a remarkable, multifaceted phenomenon that shapes who we are. It connects us to our past, informs our present, and guides our future. As we unravel the mysteries of memory, we're not just learning about the brain, we're gaining insights into the very essence of what it means to be human. Thank you for joining me on this journey into the science of memory. If you could relive or erase one memory, what would it be? Let me know in the comments below. Don't forget to like, share, and subscribe. See you in the next one.